Today, let's know how rhizobium is isolated. So today we are going to isolate one of the most beneficial microorganisms which is found in nature, Rhizobium, which is a bacterium that plays a crucial role in maintaining soil fertility and supporting sustainable agriculture. Plants require nitrogen for the synthesis of proteins, nucleic acid, chlorophyll, and many other essential compounds. Although atmospheric air contains nearly 78% nitrogen, plants cannot utilize this nitrogen directly. However, nature solves this problem through biological nitrogen fixation, and one of the most efficient agent of this process is Rhizobium. This bacteria established a symbiotic association with the roots of leguminous plants such as pea, bean, gram, lentil, soybean, and even clover. They invade the root hairs and stimulate the formation of specialized structures called root nodules. Within these nodules, the bacteria convert atmospheric nitrogen into ammonia, a form that plants can readily absorb and use. In return, plant provides the bacteria with carbohydrates, energy, and a protected environment. This mutually beneficial relationship is an excellent example of cooperation in nature. So, in this practical exercise, our objective is to isolate pure cultures of rhizobium from root nodules of a healthy leguminous plant. First and the foremost important step is the selection of fresh, healthy pink nodules. Pink nodules indicate act- active nitrogen fixation due to the presence of leg hemoglobin, a pigment that regulates oxygen concentration within the nodules and protects the nitrogen fixing enzyme nitrogenase. The selected nodules are thoroughly washed under running water to remove adhering soil particles. However, washing alone is insufficient because microorganisms may still be present on the surface of the nodules. Therefore, surface sterilization is carried out using suitable sterilizing agents such as ethanol and mercury chloride or sodium hypochlorite. This step is critical because it ensures that only the bacteria residing inside the nodules are isolated, eliminating external contaminants. After sterilization, nodules are rinsed several times with distilled water, which is sterile, to remove traces of disinfectant, which would otherwise inhibit bacterial growth. So, in the aseptic condition, the sterilized nodules are transferred to a sterile mortar or a glass watch and gently crushed using a sterile glass rod. And crushing releases the bacterial cells embedded within the nodule tissues into a suspension. This suspension becomes the inoculum for a culture. A loop full of suspension is then streaked into plates containing yeast extract mannitol agar, which can also be abbreviated as YEMA, a medium widely used for the cultivation of rhizobium. Yeast extract provides vitamin and growth factors, while mannitol serves as an energy source. Sometimes Congo red dye is incorporated into the medium because rhizobium typically absorbs the dye poorly, helping distinguish it from the contaminating bacteria that may be present. So the inoculated plates are incubated at an appropriate temperature, generally around 28 degrees centigrade, for several days. During this period, individual bacterial cells multiply to form visible colonies. The appearance of these colonies provide important clues for identification, and colonies of rhizobium are usually circular, smooth, translucent to opaque, glistening, elevated, and mucilaginous due to the production of extracellular polysaccharide. On yema containing Congo red, they often appear white or pale because they do not take up the dye readily. And to obtain a pure culture, well isolated colonies are selected and subcultured into fresh yema plates or slants using aseptic techniques. Purification is essential because mixed culture can interfere with the accurate identification and further studies. The pure culture obtained can subsequently be used for morphological observation, gram staining, biochemical characterization, and preparation of the biofertilizer inoculants. This practical not only teaches us the techniques of microbial isolation and aseptic handling, but also reveals the immense agricultural significance of beneficial microorganisms. The tiny rhizobium cells, which is hidden within the root nodules, contribute substantially to the natural enrichment of soil nitrogen, reducing the dependence on synthetic fertilizers. They actively enhances crop productivity, improves soil health, lowers production cost, and promotes environmentally sustainable farming practices. Thus, through the isolation of Rhizobium, we gain insight into the unseen microbial partnerships that sustain life on Earth. The experiment reminds us that some of the greatest contributors to the global food security are invisible to naked eye, working silently beneath the soil to nourish plants and maintain ecological balance. So we can conclude that isolation of rhizobium from root nodules demonstrates the principles of symbiotic nitrogen fixation and highlights the importance of beneficial soil microorganisms in sustainable agriculture. So by studying these bacteria, we can learn how nature itself provides solutions for improving soil fertility and supporting future food production. Thank you.